Hi guys, welcome to my YouTube channel. In this video, I will discuss about hemoglobin. So let's start. Okay. Along with hemoglobin, we also read the structure of hemoglobin and function of hemoglobin and hemoprotein. So firstly, we will read or go through the hemoprotein. First, we have to know what is hemoprotein. So hemoprotein it's a protein that contains a heme prosthetic group, and it's conjugated protein that functions in interaction it functions sorry it's a conjugated protein that functions in interaction with other chemical groups attached by the covalent bonding. Here the complexes consist of both amino acids and non-protein components. So heme containing several proteins are hemoglobin, myoglobin, cytochromes. These are the heme containing proteins, and there are some heme containing enzymes are also present like catalases, peroxidases, tryptophan dioxygenase, and other tryptophan peptidolases are also heme containing proteins. Okay, so let's move to the hemoglobin. So this is the hemoglobin, and if you divide the word hemoglobin, you can get heme plus globin. Heme, firstly, that heme is a prosthetic group and globin it's a protein. So it is the overall picture of hemoglobin, and here you can see this is the heme group, and this is the beta 1, beta 2, alpha 2, alpha 1, alpha 2 4 polypeptide chains are present in the hemoglobin. Okay. Now we can see different HB expressed during different developmental stages of humans. In embryonic stage, we can see uh, zeta 1, zeta 2, epsilon 1, epsilon 2, alpha 1, alpha 2, gamma 1, gamma 2, and alpha 1, alpha 2, beta 1, beta 2. Okay, so this is the globin proteins. The globin proteins, like here in the fetus stage, sorry, in the embryonic stages, there are uh, zeta 2, epsilon 2, and fetal, there is the alpha 2, gamma 2, and in adult hemoglobin this is the alpha 2, beta 2. It is the structure of heme. So it is a structure of heme. In this case, you can see the functional form iron in heme is ferrous, Fe plus plus ferrous form. It is a centrally placed Fe. It is centrally placed Fe plus plus. It is in reduced state. Fe plus plus located centrally in the protophoretic ring system, and Fe heme it's hexavalent. So the four bond, this bonds here and here, this four bonds linked with each nitrogen of four chiral ring. So nitrogens also contain also bonded with a chiral ring here, here, and here. There's four chiral rings are also present here, and also four bond linked with each nitrogen with four chiral rings. Fifth bond linked with proximal dishistidine F8 of globin chain, and sixth bond with oxygen. So here you can see the fifth bond with globin chain and sixth bond with oxygen. And uh, the structure of the protoporphyrin ring, that is a structure of protoporphyrin ring, is the simplest diagram. So here you can see this picture also simplify in this picture, and here you can see this Fe is centrally placed and bonded with four nitrogen, and the chiral ring also bonded with this part, these groups. So cyclic substituted tetraphyral ring structure. There is four chiral ring. You have also seen that here, one, two, three, and four. Four chiral rings are also present. And the tetrapyral ring has substituted groups in systemic manner like MV, MV, that means methyl vinyl, methyl vinyl, methyl propenyl and methyl propenyl in this manner. Again, you can see this is the three-dimensional picture of this hemoglobin. Sorry, only heme. So oxygen binding to a heme group. Now you can see it is the AFP. It is the AFP and the fifth bond with histidine F8, and another is with oxygen. Sixth bond is as it hexavalent. So the sixth bond also joined with oxygen, and distal histidine stabilizes the binding of O2 to heme. Okay, so it would clear your uh, understanding. Okay, next move to the conversion to methemoglobin to hemoglobin is NADPH plus H plus dependent. As you know, hemoglobin, there's functional hemoglobin is Fe2 plus plus state, and this hemoglobin uh, and that is this hemoglobin, when it comes with H2O2 and oxidant drug, then it comes to meth hemoglobin Fe3 plus, and the meth hemoglobin, that is Fe3 plus, also converted again to hemoglobin by meth hemoglobin reductase NADPH plus H plus into NADP plus, and it is the meth hemoglobin reductase that convert it meth hemoglobin from hemoglobin again. Okay, so let's see the structure of the structure of the globin. Before we have already uh, discussed the structure of heme. Now we will read the structure of globin. The globin protein of each molecule consists of four polypeptide chains. Two different types, alpha and non-alpha polypeptides. And in normal adults, majority of the HB is HBA type, and it consists of two alpha and two beta chains. Each alpha and beta polypeptide chains are composed of 141 and 146 amino acids respectively. And alpha chains have valine, leucine, serine in N-terminal residues, and lysine, tyrosine, arginine in C-terminal residues. And another thing is that the beta chains have valine, histidine, leucine in N-terminal residues, and lysine, tyrosine, and histidine in C-terminal residues. So now you can see the clear picture. It's a Here you can see in the globin structure. Uh, Adult hemoglobin has four polypeptide chains, two alpha and two beta polypeptide chains. And in hemoglobin, four polypeptide chains are visualized as two identical dimer. Alpha alpha 1 beta 1 is a one dimer, alpha beta 1 and alpha beta 2, they're two identical dimers. And two dimers linked to each other by weak polar bonds, that movement at interface to these two occur more freely, and two polypeptide chains within a dimer are held together tightly by ionic bonds and hydrophobic interaction, which prevent their movement relative to each other. Okay, so this is the overall picture. As you before see this here. Here this is a heme group, and this is the polypeptide chains. And this heme group, if you zoom it, then you can get this structure as you as you saw it before. Okay. So let's see the function of hemoglobin. Imparts the red color to the blood and helps to carry out the oxygen and other gases, assisting the respiratory system, and it buffers the blood pH and maintains to its tolerance limit. Source of physiological activities, active catabolites, genetic resistance to malaria, etc. And if I summarize this, then I will say that hemoglobin performs several parallel biological functions beside its basic function of oxygen and carbon dioxide transport. I mean that hemoglobin that performs several parallel biological functions beside its basic function of oxygen and carbon dioxide transport, and it performs an important role as buffer. Buffers are aqueous system that tends that tend to resist changes in pH when small amount of acid or base are added. A buffer system consists of a weak acid, the proton donor, and its conjugate base, the proton acceptor. Hemoglobin also transport the nitric oxide, and that is additional bond, nitric oxide bonds binding site is present on the beta chain, and hemoglobin acts as a modulator of erythrocyte metabolism and molecular heat transducer also. As it was about the basic function of hemoglobin, and you can also note down this normal hemoglobin functions from here. Okay, thanks for watching, and if you like the video, please hit the like button, and you can also subscribe. Thank you. Goodbye.